Okay, good morning, everybody. It's my great pleasure to introduce our speaker today. Is Susana Huelga, is a professor at the Institute of Theoretical Physics at the Yale University. She got her PhD at the Universidad de Oviedo, and later she moved to the Clarendon at Oxford University for a while. She also was a professor titular at the Universidad de Oviedo. And later she went to the University of Hertfordshire. Since 2009, she's a professor at this institute I mentioned before, at the University of Ulm. She's a leader in the field of quantum information theory. She is particularly well known for many of their works, the works her group and she has been doing in different parts of it. Including quantum metrology, theory of open quantum systems, and more. Together with Ángel Rivas, she's the author of the excellent book on quantum open quantum systems. A part of her work, which I find particularly interesting, is that on quantum effects in biological systems, as described in the thrilling book Life on the Edge by Ika Lilly and McFadden. In this field, she has made fundamental contributions which are well recognized in many publications and books. For instance, that on quantum effect in biology by her presentation today is on coherent effects in biological processes. So thank you.
Thank you. Oops. I have.
Yeah.
Because my voice today is a bit funny, but you can hear me, right? Also in the back. Okay, so as Carlos mentioned, my main line of research is the dynamics of open systems. Open systems, you know, that are subject to the influence of both coherent interactions and also uncontrollable fluctuations for degrees of freedom that are around your system of interest, right? And a few years ago, and actually I will try to explain you how the whole story evolved so that you can see the type of problems we faced and the type of methodology that we didn't have and we needed to develop to actually pay attention to processes very, very far from our comfort zone. Yeah? And those are some picosecond time scale that underpin primary processes in natural phenomena. Yeah? So in particular, the one I will focus today, simply because it's the one we have more experiments about and more theory has been done, concerns photosynthetics processes. And I've illustrated it with a beautiful example of a photosynthetic element that maybe you've seen the current exhibition at the dissect by Isabel Quintanilla, which I found a fascinating thing. If we look back, it's actually at the beginning of the development of quantum theory that the very founders started to elucubrate how far we can go with this theory that we are developing. Can actually be that, in fact, quantum mechanics, this new artifact, will have any impact in higher levels of complexity. So maybe you've heard, let's see what I can. Yes. Maybe you've heard about this book by Schrödinger. I think he was the first one to start to think more seriously about it. And in fact, the people that later unveiled the structure of DNA, it seems that they were influenced by this thinking of Schrödinger. But what I didn't know, actually, I discovered when I was looking for a cover of this book, but Niels Bohr, and can we see the date? Yeah, in 1932, he gave also a talk And you can see the paper is now reprinted in Nature, where he also explained his vision on how maybe the theory of quantum mechanics will be relevant for life. I think Bohr was much more conservative. I think Schrödinger was more adventurous. So let's see at the end of the talk on which side you agree more. So just for those of you interested by now, if you want to read a little bit of what I will call non-trivial coherent effects in biological processes, we have already quite a few references at the point of presenting some compilations of ideas, starting from 2011 up to, let's say, actually took the advantage of one of our more recent works in 2022. Okay, so I've written in here non-trivial, and this is going to be an important word in the sense that, of course, I mentioned already picosecond time scales, even below femtosecond time scales. It's not surprising that quantum mechanics may have something to say in those domains, right? But we want to explore if there is something to say beyond the formation of chemical bond or having some proton tunneling. So it's non-trivial in that sense, and as we'll elaborate later. This is the arena we're going to explore. Typically, we will be dealing with very unskillful with this. I will be interested with photo-activated processes, right, that will involve, in many cases, membrane proteins. And the common denominator is that I will be dealing with things that happen very fast, right? And that will be very efficient. So the quantum yield will be very high. There are currently evidence, stronger or weaker, right, of coherent effects in a series of processes ranging from the one we will analyze in photosynthesis, but also in the isomerization of retinal in the primary steps of vision. Also very nice work on magnetoreception that I will discuss tomorrow in the Fundación Areces, for instance. Very unexplored is chiral induced spin selectivity. Keeps busy quite a few theories and experimentalists at the moment. So this is the outlook of the talk. I've organized it in a modular manner in such a way that we can stop at different points, right? So you see that I speak quite slowly. So Carlos will ask me when is enough. So what I would like to do is first telling you exactly what dynamics are we looking at, what sort of experiments have been performed, and on which grounds these words in quotations, long-lived coherence, have starting to be plaguing the literature. What is meant by that? What is long and what sort of coherence is involved? And then the bulk of the talk from the technical point of view was trying to build some theory around it. So we're trying to put forward a theoretical framework, right, that should be able to give us an understanding of this long-lived coherence. And in addition, we don't want to make science fiction. We want to put forward a model that should be able to reproduce the different experimental data that are around. Okay, so take into account that I'm a theoretician, right? I know very little about biology, but probably we don't have any in the audience, right? So we are safe in that respect. And also I'm a theorist of a certain kind, right? So this is also an illustration to tell you how people working in open quantum systems and with a particular interest in understanding how quantum effects can remain even in a messy environment. How do they manage to actually account for the primary steps of photosynthesis? Okay, so these are the typical cartoons you would encounter if you start to look at a textbook in photosynthesis. Nature has developed an absolutely amazing machinery to be able to transform solar energy into chemical energy, into food. So of all these, we are only gonna be interested in what happens in what are called the primary steps. So the first few picoseconds, where really the task is that energy in natural complexes will be absorbed from the sun, right? Energy has to be transferred from the antenna region where these specialized complexes work, normally, for instance, embedded in the transmembrane in plants of these organelles, the chloroplast, right? The point is that energy that comes at some region has to find its way to a special destination, which has a very well-defined architecture, actually conserved across species. While this molecular network in between, depending on where the photosynthetic complex is operating, it actually may have a varied range of geometries. But you can already see in the cartoon, oops, this was, I did something. You already see in the cartoon that somehow is depicted like a migration from one of these red blobs to the other until it finally reaches the reaction center. So we're going to scrutinize indeed how this may happen, what experiments say, and how can the theory react. So for the quantum opticians, this is like the James Cummings model of quantum biology. We are going to discuss, in order to make some modeling, a very specific and probably from the biological point of view, highly irrelevant natural complex that is present in green sulfur bacteria. Green sulfur bacteria, this would be the typical habitat. So a rather horrible surroundings with very little light. And then these complexes are likely to have evolved in a manner that once a photon is absorbed, it will really, with 100% quantum yield, be delivered to the reaction center. Why is the James Cummings model? Because from the biological or the biochemical point of view, has a very, very modest size. So the typical structure we will encounter is a combination of absorbing molecules. So for instance, in this case, bacteriochlorophylls, right, that are embedded in a protein scaffold. So in the moment that you have a combination like this, then there is the possibility of having chromophores in one place or another. So one variable is going to be what is the distance in between pigments, right? What is the orientation or the dipole moments, right? Because these two parameters will then dictate what sort of talking they can have in between. And then in principle, let's say we can think of the surrounding protein as a cage holding them down. but you will see that actually this cage is far from a static structure, and is going to have a fundamental role in actually controlling the dynamics and optimizing the performance. Right. So maybe to keep in timescales, the typical intra-FMO transfer is in the range from 0.1 to 5 picoseconds. Maybe it's not pointing saying very much about the protein. Maybe for historical regions, I think it was the first pigment protein complex that was analyzed by X-ray crystallography by the three guys, Fela, Matthew, and also. Right. So what would you do as a theoretical physicist if you are given with the task of trying to say anything meaningful concerning an object like this? Well, the first thing is that the form, it appears naturally, seems to be trimeric, but actually there is no evidence of any crosstalk in between the trimers. So our basic element is going to be just one configuration with 8 bacteriochlorophylls in a protein scaffold. Okay, let's start dividing it into 2 and thinking that if some coherence is built up, then actually it's likely to be due to the coherent interaction in between bacteriochlorophylls. So let's see, this is the only animation I have, so I'm very curious to see whether it works. No. Let's see. Come on. Oh, excellent. So you see, this is rather intimidating in the sense that this is definitely what people like to call a hot, eventually room temperature, wet and noisy, right? In the sense that this assembly of bacteriochlorophylls are going to be subject to a phononic environment, right? That is introducing a lot of fluctuations in what would be the purely coherent interaction between bacteriochlorophylls. So we're going to transform this thing, right, into something amenable to do a theoretical treatment within the framework of open systems. What does this mean? This means that I define what is my system of interest, which is going to be this blob in here, that all the assembly of bacteriochlorophylls has been transformed into that, right? And then all the fluctuations, I put them around in such a way that I will try to formulate dynamical equations of motion that should be predictive, should tell me about the dynamics, and then I put them to the test to see how well they can reproduce whatever is observed. Okay, how should we think about these complicated objects, these bacteriochlorophylls? Well, really, for the task I have at hand at the moment, the only thing that matters is whether they are peacefully in the ground state or whether they have been photoexcited by the external irradiation. In the moment that there is an excitation into the system, then the whole thing kicks off. Yeah, the chromophores start to see each other, and I will model the coherent interaction with an object that will be familiar for most of you, right? So on the one hand, I have contributions that come from the side energies. This will typically be an optical transition. And then the chromophores will talk to each other by means of this coupling Hamiltonian that would allow an excitation injected in one of the chromophores, if you think of some laboratory conditions, to migrate across the network in natural conditions will be more complicated. You will have to model the external irradiation by an incoherent field. But let us concentrate in the simplest case where actually the geometry is such that there is one bacteriochlorophyll closer to the antenna. So imagine that I excited makes a transition to the excited state, and now this chromophoric network starts to interact. I need numbers to start with, right? Normally we're very used to say the system Hamiltonian is this. Well, you will see that in this context, this is a highly, highly non-trivial issue. But let's say that I now use all the experimental information, which for this small complex is actually quite vast. So we have linear spectroscopy of many sorts. I think this is absorption. Then there are also some forms of dichroism. Then we have all the values of the X-ray analysis. So let's say I have enough information to actually put numbers to what would be the energies of all the bacteriochlorophylls, and also at least an estimation of what the possible coherent coupling between them is going to be. Moreover, people have done more sophisticated experiments where they use now sequences of laser pulses, right? In such a way that now you have the ability to control what is the spacing in between pulses that will create coherence, and then you should be able to detect it. I think here you have on-campus experiments already doing PAM-probe, and just had a talk of, forgotten his name, Ferry, yeah, is gonna actually be able to soon implement femtosecond spectroscopy. So maybe I'll give you an indication of what these type of experiments are, because those were the ones that capture the attention of the theoretical physics community. Nobody was interested in the linear spectroscopy of this type of complexes or photosynthesis in general in the theoretical physics camp. But I believe that this was one of the really pioneering experiments when Tobias Brixner was a postdoc in the group of Graham Fleming in Berkeley. So let me read out. They say we monitor the excitation energy transfer as a function of time and frequency, and show that excitation energy does not simply cascade stepwise down the energy. So they essentially challenge how this is really happening. How should I think about it? Is the energy jumping from chromophore to chromophore? Is energy being delocalized as a result of the excitonic interaction, and then have a considerably more complicated transfer paths? Is it that excitons do form with certain degree of delocalization and then thermally cascade? So this is what we want to analyze. I think I will not enter too much into this because it will probably take me ages to explain it. But let me go fast. Sorry, I hate when people do this and I'm in the audience. But okay, essentially what you try to do with this multidimensional spectroscopy is being able to create correlation maps in between the frequencies at which you excite and the frequency to which you detect, in such a manner that when you vary the time in between pulses and you Fourier transform at each time, then effectively what you are able to create is the possibility to follow up the whereabouts of all the energy landscape. So you can not only witness the possible formation of excitonic states, that you can argue you can also see them with linear spectroscopy, but you actually are able to see the fingerprints of the dynamics. Let me go to the bones that I think we will all understand in terms of Ramsey interferometry. So the main idea of this type of experiments from the point of view of a theoretician is that there are experiments where you can create coherence. Here I'm considering the simplest case. Imagine that I only have two chromophores talking to each other. So then I have a ground state, and then I have, so bad with this pointer. Then I have two excitonic states. If the illumination is weak enough, then the probability to have two excitations at the same time is very small. So we can remain confined to the, I was taught that Harosh lost a lot of audience, so I'm not too bothered. It's only one at the moment. Okay. So we have two excitonic states. Imagine that why do I lose the point? Imagine that the splitting is this capital omega, right? And then we have an essentially optical transition down to the ground state. So imagine that now you have the possibility of creating, by means of a Bio2 pulse with the right timing, you have the possibility of creating a coherent superposition of these two excitonic states, and then you probe them, right? If you are in a completely isolated environment, if this whole evolution will be truly unitary, then you know that you have the typical interference signal, right, with a sinusoidal profile. You would be able to witness full population inversion. Well, if there is noise around, this will be mapped out in the damping of the observed oscillations. So here you can understand where the wording long-lived coherence comes about. Because what experiments tell you is that the lifetime of a typical ground-excited superposition is of the order of femtoseconds. However, it seems that the non-linear, you see that I'm being very cautious in what I say. It seems, or there are indications in the non-linear response that they see signatures of oscillatory behavior that is going well beyond the femtosecond time scale. So the coherence is long and maybe of electronic origin, right? In the sense that exceeding by quite a bit the order of magnitude of the typical dephasing rates that they are observed at the level of a single chromophore. Okay? So why? How is it possible that in this type of systems and even at room temperature, this may be happening? So let me emphasize that idea again. The interpretation that Fleming and his group and others did, also with other type of complexes, is that there seem to be oscillatory signals of possible electronic origin, right, whose lifetime extends in the typical time scale of an energy transfer process. So this is what we would call a non-trivial effect, right? In the sense that on a time scale that's something relevant, like the energy transfer from the antenna to the reaction center, right, seems to show signatures of the underpinning of a coherent interaction in between chromophores. Again, a bit of history. Maybe these were the most celebrated experiments, but there are by now many others for many people, including some by Nick van Hulst in Barcelona. So From the theoretical point of view, you can also think of evaluating, even in a numerically exact manner for some small complexes, what the spectral response would be. But I again skip this. This is only that if you feel tempted to compute it, it's clear that you have to evaluate a third order polarization. But let's move back more to the phenomenology. Right, so then we have the following situations. There seems to be this type of experimental result, and therefore a very interesting prospect, right, that may be a quantitative relation in between the fact that we have coherent effects in an interesting time scale and the possible way these complexes function. Is there any connection actually in between coherence and functionality in biology? This was, I think, the catch to get the information, the quantum information community, in particular the people interested in open system, to pay attention, right? And then also, if you want, also for the open system community, it was a challenge to really being able to see whether the type of tools we've been deploying for many other studies, right, actually can also be helpful in some manner here. Okay, so two tasks then. Try to understand from the microscopic point of view what is going on. What sort of microscopics are here that allow me to make some predictions on whether or not this coherent behavior will happen, whether it makes sense to indeed expect that it's not just a purely dissipative phenomena. I would say that we have quite a few things to say here. And then if you want, the more challenging thing will be saying, okay, once you've showed that indeed the physics seems to indicate that this is feasible, can we think of any task in biology? Maybe to be more modest, one would say in molecular biology, that we can show that is coherent assisted, in the sense that either this coherence is built up and the process happens with some efficiency, or if it's absent, really doesn't happen, right? So very much like if you want to perform perfect quantum state teleportation, you need to share a bit of entanglement. If you don't have it, bad luck, you cannot teleport. Well, as you can imagine, the second part is entirely open for the moment. So why our group started to be interested in this case? Because many years ago, right, we were working a lot in the context of implementing quantum information processing tasks, right? We were trying to explore whether there were different routes to try to implement quantum error correction, a task that even nowadays is immensely costly. So then the idea was, can we actually exploit the noise that is in the system for something advantageous? Well, nowadays, this is mainstream physics, right? Whole field of reservoir engineering. Let's say we know a lot of microscopic mechanisms that actually need the combination of both noise and coherence to be able to have entanglement quantum correlations that are lived in the asymptotic limit. It's less clear so that the combination of noise and coherence can be advantageous in the transients. And this seems to be what is happening in the biology context. Okay, so from bio to theoretical physics, this is the type of analysis we're going to be performing. So we are going to describe the interaction between chromophores with the very same Hamiltonian I told you about before. And then the different levels of complexity are going to come in the modeling of the chromophoric network with all the surrounding vibrational degrees of freedom. So typical onion-like construction. We will start with a modeling where actually put all the vibrations under a Markovian umbrella and see how far we can go. And then we will start to sophisticate the system. Because actually, if we look at the parameters, the initial parameters that we can input in our Hamiltonian for the type of interaction strength in between chromophores and in between the local sites and the different vibrations, and in particular the coupling with their own intramolecular vibrations, we see that it's a no man's land. So the typical clear separation that you would like to have in order to start doing a perturbative treatment of the phonon bath, they are not there. But let's assume that they are and see whether we can say something meaningful. This is an idea that I want to stress from the very beginning, right? So if you talk with any experimentalist doing spectroscopy with these complexes, they will tell you that, of course, the spectral response is full of oscillations. In fact, you will have to fight not to see any. So there is no doubt there is coherence in these dynamics. But the point is that coherence may come from, let's call it a boring one, from oscillations in the ground state and some vibrational modes. This is something we are not interested. We are interested of something that involves the excited state manifold in a non-trivial manner. Okay? Moreover, it doesn't matter how nice is the theoretical model you put forward, we have a vast amount of spectroscopy we have to account for, right? So we need a model that is consistent also with everything the people have been measured, linear and non-linear spectroscopy. Okay, first attempt, what would you do? Saying, okay, I go for making a Lindbladian analysis of the dynamics. Phenomenologically, taking into account what are the typical lifetimes that are being measured, I simply write down a Hamiltonian contribution that is going to be very inefficient from the point of view of transfer. Easy to understand, right? Imagine a network of two-level systems coherently coupled, able to exchange excitations of some form, and completely isolated. Well, depending on the strength of the interactions, it's actually very likely that you will have some superpositions that if your aim is to transfer energy somewhere else, they don't have any overlap with this somewhere else. So you have a fraction of the energy on limbo there. So at that respect, coherence doesn't seem to be very useful. But let's see what happens if actually, and in fact, you can do an analytical calculation saying the probability that you transfer efficiently is actually inverse to the number of networks. In the moment that this grows, that's anything but conveying energy to the right place. So can noise help? And I think you can bet that it will do, right? So the simplest model you can introduce is local dephasing, simply because we know from the experiments that locally is the first thing to attack. Actually, ground excited state superpositions, I told you that they live femtoseconds. So we introduce possible loss of energy happens on a much longer time scale. So let's see what happens when I have local dephasing. Well, you see that the perfect network was pretty bad for conveying energy. You start to introduce dephasing, you increase dephasing and see that things have actually improved. You may be able to transfer in a few picoseconds with the parameters for FMO. So definitely noise is helping. It's helping in the sense that will allow to bridge energy gaps that were not very favorable initially, with the landscape dictated by the Hamiltonian. And moreover, very much like in a Mach center interferometer that is completely balanced, it will open paths that were initially forbidden. So then the typical behavior we will have for what would be, if you want, the efficiency of the complex as a function of the noise strength is highly non-monotonic. You have a value here, a broad plateau, as one would expect for a natural complex, right? Where actually the combination of this local dephasing together with the Hamiltonian interaction allows you to efficiently convey excitations. If noise is too little or is too much, then the operation of the system is hindered. Of course, these things can happen, and this is a nice illustration of this type of prediction in the context of energy transfer along an ion chain. So no doubt that we have this type of quite intuitive way of local dephasing to assist energy transfer. But will the theoretical model we've been putting forward being able to reproduce all the spectroscopy? Because otherwise, we put it in the chapter of science fiction with respect to the biological point of view, right? Have a look at the typical spectrum of the fluctuations. Is the last thing you want to see if you want to do a microscopic and ab initio derivation of master equations of Lindblad form, right? So look again at the initial estimation of the different parameters that will govern whether you are safe or not doing the type of second order perturbation that you effectively do in the Lindblad treatment, and the answer is no. So the whole thing has to be more sophisticated. Despite the general spirit, I admit, is qualitative, it will persist. So it seems that these pigment protein complexes operate in a regime where they really benefit of a little bit of coherence and a suitable combination with the fluctuations around. But the role of the fluctuations is much more sophisticated than simply, effectively, the model I've done before just relaxes excitons thermally. If you transform this local dephasing, how it would look like in the basis of the excitons, they provide you the mechanism for excess energy to be dropped into the vibrations and just the excitonic ladder to go down to the lowest energy exciton. But the role of the vibrations, as we will see, is different. And moreover, it will be essential to really clarify what is the nature of the oscillations that we are observing, in the sense of arguing that the possible electronic coherence that will build up is really residual and gone in a femtosecond time scale, and then everything else just being vibrational motion of the modes, or whether there is mixing. Because you can say, okay, this is actually bad news for deriving simple Markovian master equations, but it's good news for actually having now an interacting system with a lot of hybrid entities will build up by Brownian components, given that I have on the one hand an electronic coupling trying to talk to them between chromophores, and also a strong coupling with some vibrations. The typical profile of the spectral density sources, generally a combination of molecular dynamics and also measurements done at low T using fluorescence line narrowing. Let's say that we have at least an educated guess to build up the type of spectral density. Actually, the data I showed you before of the linear spectroscopy and the parameters that are fed into the Hamiltonian are typically computed by means of making theory that has as an input a certain spectral density. Well, the type of spectral density that was used will have the form that we will discuss in a moment. What I'm saying, I told you at the very beginning that the issue of starting with a certain Hamiltonian is highly non-trivial. And this is going to be very relevant in the discussion of the nature of the oscillations. Why? Because the numbers I told you are not computed with a true spectral density, but with a highly coarse-grained one. So then imagine if I do the fitting now with a different spectral density and I'm anticipating results, then things can change. If the parameters of the Hamiltonian change, all of a sudden the coupling strength in between bacteriochlorophylls actually is larger, then the electronic component in the oscillations could be enhanced. Right, this I've already said. So let's move to the next order of complexity. What I'm going to do, very much motivated by the intuition that if I'm interested in energy transfer, and then there is some possible non-trivial role of the phononic environment, I want to go beyond Markovian master equations, then really what should matter is what sort of vibrations I do have in the vicinity of my suspected excitonic gaps. So let's start dwelling into the possible nature of the microscopic interactions that may collaborate in having a long-lived excitonic coherence. So then what we did was forgetting about reduced equations of motion. I will always be ransom of the type of approximations I have to introduce. Wow. So I will try to do a numerically exact analysis. Okay? No perturbative analysis. But then, of course, I have to be modest and say, for the moment, I can forget about FMO. Let's see what I can model numerically exact, the simplest structure that would be made up of only 2 bacteriochlorophylls. So then in the model I'm going to show you, we only consider 2 excitonic states that correspond to the 2 lowest excitons in FMO. Also because those are the ones expected to yield to the strongest signals. Right, so this is what we did. Quantum opticians in the audience can already imagine why this is going to be advantageous for coherence to be preserved, right? Because if I have vibrational modes in the vicinity of the excitonic transitions, right, what happens is that when I look at the excitonic dynamics and I see what this type of profile will do, it would be saying, okay, the rest of the fluctuations, the ones that are far resonance, they will effectively try to decohere these superpositions, but at the same time, I have something quasi-resonant with the excitonic gap that is trying to revamp the coherence that is lost. So this is very much like dephasing while simultaneously being coherently driven. So then, indeed, we did a numerically exact analysis. I think I will make another jumping here. Oh, well, no, I showed you this cartoon. Even just more pictorially, right? The role of these vibrational modes is giving you a push in the right moment, right? So off, we keep them oscillating. So yes, if we do now, we use tensor network type of numerical simulations that you also use a lot in this group, right? To do a numerically exact analysis and see how does purely excitonic coherence behave. And indeed, unsurprisingly, these are results at 77, but probably more interested is at room temperature. So here we're making a theoretical calculation of how would a measure of the electronic coherence behave, dependently on whether the background fluctuations contain or don't dashed a quasi-resonant vibrational mode. So that would be an argument to think that in the presence of these structured fluctuations, indeed, we have built-in mechanisms that actually conspire to keep coherence alive. And indeed, you can do 2D calculations and see whether your predictions have any resemble to the actual ones, what we indeed verified. You can see whether some artificial systems, less complex, that are real natural pigment protein would be accountable for, and within theory for specific experiments. But nevertheless, we are back to square one, because you see, we put one of the modes that are resonant with the excitonic transition, but we have many other people in the party. You can say anything that is off resonance shouldn't do too much damage, but this is also naive. Because, okay, they are very far, but there are many. So, last chapter, I will jump again. I think I overestimated my capabilities as talking fast enough. But let's say that we deployed all our arsenal, everything we have, more and more sophisticated tensor network analysis, right? If anybody is interested, we can talk later. But I go to tell you our more recent work. Numerically exact simulations with a full Monty. Actual spectral density. One complex, that is the real one. This is present actually in cauliflower. And the second one is not the full complex, but it has the interest of being the special pair of the bacterial reaction center, which is the structure that every biologist will tell you that is the most likely to have been optimized in nature to perform charge transfer. Right. So this is a numerical exact calculation of absorption spectra of the complex WSPC that has been evaluated. We have the green dots, the experimental data, and then you see what is the numerically exact calculation when we include only 20 high energy mode, 40, 55. And you see that we can reproduce with all the 55 the experimentally measured absorption, but the Hamiltonian has to be renormalized. So the debate on the nature of the oscillations is, I would say, back in. We can argue that the whole inclusion of the spectral density will introduce notable changes in the excitonic dynamics. You can say that the excitonic dynamics is not accessible experimentally per se. What you have is the spectral response. It does, of course, manifest in the spectral response. So this is the state of the art. All the previous experimental fittings, all the theory to do the experimental fittings have considered simplified forms of the spectral density. So when the whole spectral density is introduced, then, and this is the way, right? Theoretical models are fine-tuned. Then, depending on the complex, this is also not going to be a universal feature, would experience renormalization. So for instance, the estimated coupling strength in the special pair is likely to be higher. So therefore, what is the nature of the oscillations we observe in the spectral response? I would argue that it's mixed. It has both an electronic and vibrational component. And if the parameters are right, the electronic component may be more significant than thought. Okay, so it may be time to start packing up, right? So at the risk of repeating myself, it's difficult in these systems to connect the dynamics that is not directly accessible. We cannot tomography the dynamics in the system. So it's difficult to make a one-to-one correspondence in between the spectral response, unless you would do, instead of two-dimensional, 50-dimensional spectroscopy, right? So eventually reconstruct the whole dynamics. Then it's highly non-trivial to connect the spectral response and the dynamics. And this is precisely what has made this debate of how electronic the coherence is for quite a few years. We simply didn't have all the numerical tools to actually properly address these systems. So putting these two lenses together is complicated, but now, actually, this is now published last Friday in PRL. We can compute exactly the absorption spectra of FMO. So actually, the 8 bacteriochlorophylls. What is the, don't tell you how we did it. What is the picture? Well, it seems that indeed the bibronic model, when we have this multi-mode actually mixing of electron and phonon, seems at least able to provide some light. You can argue, okay, is there something beyond a matter of pride on using the potential of tensor networks in actually clarifying what is the nature of these oscillations? Well, this is something we will not discuss, but we can formulate the problem saying, I have a figure of merit, the power that a certain device can develop, and map him out to this situation where I have a network of chromophores operating between solar irradiation and a cold background of the phonon bath. I'm trying to do some task. Well, that's a nice scenario to do quantum thermodynamics and actually show that the power you can deliver is a function of the coherence that can be built up So that would be an argument in the direction that maybe we can connect the coherence that the dynamics will build up with some enhanced functionality. But that would be for some other talk. And see whether I can jump to the acknowledgements, probably not. And I don't want to bore you with back and forth. I go back here. Okay? So what I told you ran for 10 years or so. So you can imagine that there was quite a few people involved. So have the names in a slide number, whatever. But it would be important. In particular, a very big thanks to a friend of both Luis Quiroga and myself, that was Felipe Caicedo Soler, that was in our group and passed away in the Alps. So this is a tribute to him as well. Thanks for your attention.
Okay, thank you very much for this very nice, interesting talk.
Thank you.
Questions, comments, criticisms?
Yeah. Sure.
Yeah, thanks a lot. I wanted to ask about the coarse graining, kind of how do you do it, or where does it come from? What's the argument of doing it?
So you mean the one perform in order to be able to compute? Yes. Exactly, right. So then the idea is that you coarse grain it on the basis of having the two-time correlation function modulo, assuming that the environment is Gaussian by linear interactions, etc. Right? So instead of making a fitting of the exact spectral density to the total, to be able to have the complete correlation function, what you do is taking a duration that corresponds to the time scale of the response you are going to quantify. So for linear spectrum, will be shorter. If you want to account for two linear spectroscopy, you have to extend it. But it's as simple as that, but actually showing that this can be done systematically, right, requires some tricks. Actually proving the formal equivalence in between certain unitary and certain dissipative transformations, right, evolutions, proving that actually if they have the same single and two-time correlators, then at the level of expectation values, the dynamics is the same. Right.
Hi, thank you for the nice talk. It's not a question, but is that to my understanding, usually there is the debate between whether there are coherences in this efficient transport of excitation, that the experiments are performed under illumination of a coherent source. So these two experiments are made with laser sources.
Right.
And I was wondering if you could comment on that.
Absolutely, that's a very pertinent comment, because indeed, one question is, could these complexes support coherence under the right experimental conditions? Maybe you can argue that this could be an answer. And a different one is, can you from that extrapolate to say that in natural conditions with incoherent irradiation, right, this is going to be relevant? We cannot ensure it at all, right? We've done Mickey Mouse simulations of really injected excitation instead with laser light with an additional dissipative term. And there you see coherence being built up. But I wouldn't risk in arguing in the positive. Also in the sense that doing a numerically exact modeling of the absorption is very complex. Again, this is something Luis would definitely agree with me, right? To really model microscopically how the absorption of thermal light will happen, yeah? So nobody has done simulations that combine numerically exact absorption and numerically exact propagation. In fact, Paul Broom, maybe you know his work, he's somebody that has argued very powerful, saying, oh, this laser thing then doesn't have any relevance for actual biology. May actually be right. And he has tried to do some exact simulation of absorption. So it'd be very good to combine his input for the initialization of the system with our tools for actually propagating it and computing the spectral response. Yeah, thanks for the question.
More questions?
So my question was more like philosophical. You mentioned several times this idea of reproducing the experimental spectra, and then you make your tensor network models more and more complex with more and more parameters. Maybe you reach the point where you actually your model is not giving you any insight into the problem anymore, no? I mean, we physicists like simplified models and universality. Biologists don't like that, no? Biologists like to know, understand that particular chlorophyll or whatever photosynthetic system.
This is true, and actually one thing that initially caused a lot of frustration to us is that, let's say, some people that do theory at the level of being embedded in an experimental group, they do some theory for this experiment, they do another experiment and they change the theory. I'm sorry, we need to have, this is why I insisted that we need okay, maybe this is a problem of how we're trained, right? But we want a general thing, right? If I input this system, I have my theoretical description, off I go, right? And my only proof is what you've measured. But I cannot tell a different story. If you do something then has changed, then you cannot tell me that they are comparable. So this is relevant. We need a theoretical framework that can account for all the spectroscopy. You can say pointless, but then—
No, I didn't say that.
I mean that you can say maybe given the complexity of these ones, you are not be able to go and build it down to the level of Hamiltonian description with some system environment. And you can even say, which guarantee do you have that actually is modelable like a bosonic path? Maybe non-harmonicities, who knows, right? So, but you can see it a little bit like discrimination with Bell inequalities, right? At least then you can put out of the game the theoretical frameworks that are not fit for purpose. They cannot reproduce the spectroscopy out. Okay? So I think this will allow to refine more and more the underpinning theory. And I think definitely clarifying whether indeed or not, the spirit of the previous talk, at least these systems support coherence. Because then they can also be building blocks, you know, for hybrid structures where maybe we modify, we put them closer together than in nature. Okay.
Can you comment on other biological processes where this kind of noise-induced coherence is also present or possible?
While one is the isomerization of retinal, right? And there is clear that in the process there is a conical intersection that definitely is building up again Byronic coherence. And there are, we call it numerically exact, but let's say some refined theoretical models, right? And then another one that is quite interesting, and that's definitely involving a true biological system at the level of the modeling, is magnetoreception. Because you know that migratory birds are actually able to sense the orientation of the magnetic field, right? Well, it seems that the underpinning mechanism is actually a spin-dependent reaction that actually depends on coherence building up between electron spin and electron nuclei. Yeah, of course, experiments have done now at the level of how do they say, the biologists, they express the protein, right, in vitro, and they have checked the magnetic sensitivity of cryptochromes present in the retina of the eye. And then the next step will be deploying quantum technologies to be able, with maybe MD centers, to make in vivo measurement. If that's done, right, that would be a very, very nice, probably conceptually perhaps more clear-cut of coherent effects in a true biological system.
Okay. Just to be recorded.
Thank you. Really nice talk. I was wondering about how important or how relevant is how you populate the phonons. As I mean,
Right.
you could.
No, here I would say that that would be less controversial than the issue of the illumination, in the sense that there is thermal. Whatever the thermal temperature is, that will dictate the population of my modes.
True,
Mm.
but I mean, you could think of matrix where you could raise the Debye temperature so high, such that you would enforce that you're no longer obeying a Maxwell-Boltzmann equation, and there you'd add some color to the noise, right?
Oh, yeah, actually, you've seen the form of the spectral density. Yeah, so that's definitely, let's say, if we make the assumption, right, that I can model the phonon environment as uncoupled harmonic oscillators, so I don't want non-harmonicities. The interaction is bilinear, right? And the system is initialized in some Gaussian state, then I have the guarantee that the spectral density from the point of view of the response of the system encompasses everything, and that's temperature dependent. And in general, this will lead to a highly, highly colored dynamics.
Thank you.
Yeah.
Anybody else?
I wanted to ask exactly about the kind of, how to say, another way that I guess a lot of people would naturally try to approach the system is with molecular dynamics and quantum chemistry calculations, where then you would capture exactly the enharmonicities in the surfaces naturally, but you probably would lose a lot of the vibrational resonances because you don't have quantum vibrations. So
I mean, and you see that the input of the correct spectral form is essential for the tensor network machinery to work. So normally, these inputs tend to be mixed, right? So that there is some part of the frequencies that are well resolved experimentally, and then there may be some additional patches that come from molecular dynamics. But this is indeed very active and absolutely essential for things to move forward. So Benedetta Menucci, for instance, is one person working a lot on that. Yeah.
Last question. Okay.
Oh, there was a pair creation.
They are entangled.
Thank you for the nice talk. First of all, I'm wondering if you are talking about exact time dynamics, how much time does it take to calculate it? I assume it's not seconds.
You mean really the
Doppler calculations which you were mentioning?
Okay. Let's say that no, there are no seconds, neither hours, right? Some calculations have to run for more than a week. Yeah, but I don't think that we are really optimizing maybe. Now we have our own cluster and trying parallelizations and so we have now some people in the group that do serious numerics, right? So that is something that I trust that it will be improved. But there is also a very, very clear horizon telling you the complexity you have, you can go, right? So we will never be able to simulate in a numerically exact manner the transfer in arrays of many of these complexes, right? Because I told you FMO is a little bit of a curiosity biology from the biological point of view. So very interesting computationally would be able to do numerical simulations that can cover large time scales with extended systems in such a way that we maybe will use tensor networks for some parts, then will come other type of less expensive routines and so on. Yeah.
Thank you.
Okay, so I think it's time to acknowledge again the
Thank
talk
you.
by Susana.
Thank you very much.